Hello, and welcome to this lab number 8 overview for renal function for Bio 206 Human Physiology Lab. This version is going to be the remote version of lab number 8. As always, we have a set of objectives for these labs that correspond to what is also available on the lab handouts that are available on Canvas. So basically, for this kidney function lab, we're going to be looking at renal handling of substances and basically kind of the concentration of materials that are found within the urine. And so what we need to think about is how is it the materials are going to get into the urine, basically be excreted from the body. And so kind of what we're going to be measuring in lab, or at least measuring it in an artificial setting, is to take a look at the quantities and the quality of the solutes and the materials that are excreted from the body within the urine. Now, in order for it to be kind of voided from the body, to be eliminated from the body, it needs to go through the processing that's going to be occurring within the kidneys. And this is, again, going to correspond to what's being covered in the lecture. And so we're going to have blood coming in through that afferent arterial. It's going to go into that glomerular capillary bed, and then some materials are going to be filtered out through the podocytes, kind of wrapped around it from Bowman's capsules. You've got the fenestrated capillaries here within the glomerular capillary bed, so that, basically the blood cells and really large substances are going to be trapped within the blood system here. And then those materials that are small enough to fit through this filtration gap are going to be able to be filtered into the raw filtrate, into the raw urine here at this point, going into the start of what would actually be the urinary space, and then going into the proximal tubules over here. Now, as these materials are passing down through the tubule systems, the uriniferous tubules, the proximal convoluted tubule, the loop of Henle, and the distal tubules, we're going to look at the fact that some of these materials are going to be modified. And it's going to be modified in that some of the materials are going to be recognized as being important within the body, that need to be maintained within the body. And it's easier physiologically to kind of reclaim those materials, almost like a recycling of these materials, but to reabsorb those materials from the raw filtrate, bring it back into the connective tissue, get it into the bloodstream, and then carry it away from the kidneys. And so we're going to be removing those materials, those solutes from that raw filtrate through the process of reabsorption and decreasing the amount within the urine. At the same time, there may be other materials, or in some cases the same material, generally other materials in most cases, where we're going to look at secretion occurring. And so we're going to look at the fact that we're going to have cells kind of lining the uriniferous tubules that are going to be actively moving materials from either the bloodstream and the tissue space into that processed, or at least the developing urine in there. So the total amount that is going to be excreted is actually going to be the amount that it's going to be filtered minus the amount that is reabsorbed, or almost recycled within the body, basically brought back into the body proper, bringing it back into the tissues, plus those materials that are being secreted. And so we're looking at the renal handling of a substance as being that combination of filtration, reabsorption, and secretion. And so if we take a look at renal handling of different substances, what we can see is that for some substances, it's going to be filtered out within the renal corpuscles, within Bowman's capsules, but it also could be secreted. And so actively move from the bloodstream into that urinary space. And so a material that is filtered and secreted, but not reabsorbed, would be things like our hydrogen ions, again, kind of contributing to kind of the acidity within the environment. We can have materials that are going to be filtered and partially reabsorbed. So in this case here, we got substance X in the middle. And so it's being filtered out within Bowman's capsule in the renal corpuscle up this portion here. A portion of it is going to be reabsorbed, but some of it is still going to end up within the urine. And so things that are filtered and partially reabsorbed would be things like sodium ions and to a certain extent urea as well. We can look at some materials that under normal circumstances are going to be filtered and completely reabsorbed. And so this is like glucose. And so glucose within the bloodstream is a small enough molecule. It's able to kind of squeeze through that filtration barrier, get into the raw filtrate. But under normal circumstances, basically, if an individual is not experiencing diabetes where they have an abnormally high level of glucose within their bloodstream, the amount of glucose that is going to be filtered into that raw filtrate can be reabsorbed within the uriniferous tubules. If the blood glucose levels are too high, we end up with too much glucose being filtered in it, and it kind of is too much that can be, basically not all of it is going to be reabsorbed, and that's when we're going to start to see urine appearing, glucose appearing within the urine, which is characteristic of patients with diabetes. Finally, and we'll have an example of this in one of the questions in the lab, we can see materials that are going to be filtered, but neither reabsorbed nor secreted. And so basically what is going to be filtered out here in this process of kind of going from the glomerular capillary through the filtration barrier into the urinary space as raw filtrate, materials such as creatinine will not be reabsorbed and will not be secreted. And so the filtration rate is going to correspond to our glomerular filtration rate is going to correspond with our excretion rate. So if we take a look at this, and again, another example similar to the creatinine that is filtered, but not, it's not reabsorbed and it's not secreted, is going to be inulin. So inulin is going to be filtered, but not absorbed or secreted. And so we can measure the concentration of inulin within the urine and use that in relationship with the concentration of inulin within plasma. And in doing so, we can come up with that filtration rate of the inulin. And because the filtration of the rate of inulin is going to be equal to the glomerular filtration rate, this gives us kind of a very good measure of the kind of activity level that is going to be going on within the kidneys, the glomerular filtration rate. And again, we'll have an example of this in the questions in lab this week. Finally, and again, because we're in this remote environment, emphasizing it here, the study guide for lab exam number 2 is available on Canvas. So if you go to the module for lab exam number 2, you'll find the number 2 study guide. And again, lab number 2 is going to correspond to the questions and activities associated with lab number 5, 6, 7, and 8. So the last 4 labs, the second half of the lab. Taking a look at what we're going to be doing this week in physiology lab and what you're going to be doing kind of remotely for physiology lab this week. Same format as what we've had before. And so we've got a Google document for the lab report number 8. We've got the instructions as a PDF as well, but download the template either onto your Google Drive if you're going to be using Google Docs to edit it, or download it to your computer and use Microsoft Word to edit it. Ideally, use the naming convention in case we need to download it. And in the screenshot, you can also see some of the data that we have for the lab this week. And so what we've got are going to be test strips that have been created. So instead of having to go through and dip the sticks in the test strips in the urine itself, we're going to have it basically the results for you. So in the activity, you're basically going to do urinalysis, test strip, and then analyze the results by looking at the pattern on the different, basically the little colored squares on here are going to correspond to the different tests. And so what we would like you to do is watch the YouTube video, how to read a urine test strip. And then we've got 4 samples for you to work with. And so we've got 1 normal sample and then 3 unknown samples that you need to be able to kind of analyze. So you take a look at the colors on the test strip in relationship to the standards that are going to be present. And then based on what's available in the lab handout, come up with a possible diagnosis for the individual that provided the unknown sample of urine. Once you've completed your lab report, again, you can submit this through Canvas. So again, the submit lab report number 8 here, renal function here. So you click on that and be able to upload that directly into Canvas. As always, your lab instructor may have additional information available to you. And so please see if they have any changes or updates for the lab for this week. As always, if you have any questions, please do not hesitate to reach out to me. Thank you and have a great week in lab day.